بسم الله الرحمن الرحيم، والصلاة والسلام على خير خلقه محمد وعلى آل بيته الطيبين الطاهرين. المحاضرة الأولى entitled Classification of Bacteria, prepared by DR.IHSAN. The learning objective of the lecture: definition, define the taxonomic model, explain grouping of bacteria, determine the classification system, and clarify the taxonomy technique. The aim of this lecture is to determine the importance of bacterial taxonomy and the distribution of bacterial agents within the group and the cluster, and define the specificity. This figure illustrates the 5-kingdom system put in by Carl Lewis in 1975 that distribute the organism in the earth into 5 kingdoms: Monera, Protista, Planta, Fungi, and Animalia. Then another system was appeared in the science and known as a 3-domain system that include domain 1, bacteria, and domain archaea, and domain eukarya or eukaryotes that distribute all living organisms in this branch. The taxonomy is the study and the grouping of organisms. There are 2 taxonomic models used for a taxonomy or a classification of microorganisms, and especially bacteria, either a numerical taxonomy or a phylogenetic taxonomy. The differences between these 2 systems according to weight of character, multiple techniques, and use of computer. In a numerical taxonomy, we use a huge amount of technique that all technique has an equal weight to another technique. And this taxonomy system is depend on a morphological character and use a computer in the analysis of data. While in phylogenetic taxonomy, we use a morphological and a genetic or molecular technique used to classification of bacteria, and each technique has a weight that differ from another technique. 3 separate but intercalated areas that use to reach to taxonomy. First, identification, process of characterization organism, such as a morphological and a biochemical test. Secondly, classification, that means arrangement of organism into groups, such as arrangement of staphylococci to a gram-positive cocci, streptococci to a gram-positive cocci, bacillus subtilis to a gram-positive bacilli, spore-forming, etc. And third, a nomenclature is assignment of a specific name that I give a scientific name for each bacteria. The scientific name for each bacteria must begin in a genus name started with capital letter, and then a species must be written in italic or underline under the scientific or specific name. The taxonomic classification scheme for bacteria may be found in a very important reference known as a Bergese Manual of Systematic Bacteriology. In Bergese Manual, bacteria are divided into 4 divisions. 3 divisions consist of bacterial cell, and 4th division consists of archibacteria or unusual bacteria. Each division is divided into class. Archibacteria. Archibacteria include 3 groups. First, the methano group. The methanogenes, strict anaerobe, that produce a methane, CH4, from carbon dioxide and hydrogen, example methanococcus. Secondly, extreme halophile, which require a high concentration of salt for survival, such as a halococcus and halobacterium. Thirdly, thermoacidophiles, which normally grow in hot acidic environment. Actinomycetes. Actinomycetes are filamentous bacteria. Their morphology resembles that of filamentous fungi. However, the filament of actinomycetes consists of prokaryotic cell. Some actinomycetes resemble mold by forming externally carried asexual spore for reproduction. Filamentous has a high guanine plus cytosine content and gram positive. The ratio of guanine and cytosine content is between 63 and 78%. This is a figure of actinomycetes. The second group of bacteria is a cyanobacteria. The cyanobacteria have a typical prokaryotic cell structure and a normal gram-negative cell wall. They range in diameter from about 1 to 10 micrometer and may be unicellular or form a filamentous. They have a chlorophyll and carry out oxygen-producing photosynthesis, much as a plant and eukaryotic algae do, as a photosynthetic bacteria. The classification method for all bacteria is depend on two phenomena or two key. The firstly, dichotomous key, widely used, based upon a question, each of which has a 2 possible answer, eventually get to identification. Or secondly, cladogram are used to show a degree of evolutionary relationship. This is a dichotomous key that begin in one answer, have in one question, have a 2 answer. One question lead to 2 answer. One question lead to 2 answer. Gram reaction, positive, negative. Negative, glucose fermentation. Yield acid or not yield acid. Motile at 37 centigrade, yes or no. Urease hydrolysis, yes or no. Indole producing, yes or no. While other phenomena of hierarchy is a cladogram. The cladogram is used essentially for a numerical taxonomy. That means each taxa or a genera or a species has a weight of character, and finally each organism give a total of character that give a similarity between each of the genera. So the phenomena and finally appear as shown in this diagram. Taxonomy. Initial identification of microorganisms results in their classification. Based on evolutionary relationship, identification of microorganisms, in particular environment, remain important, such as microbial contamination can spoil food. Another example, identification of microbe present in a clinical patient is important in determining treatment. Prokaryote identification. There are various techniques are employed to characterize and identify microorganisms, divided into two levels: phenotypic characteristic and genotypic characteristic. In phenotypic characteristic, there are many techniques used to identify the microorganism and characterize of microorganism: microscopic, morphology, metabolic differences in biochemistry, serology, and fatty acid analysis. While in genotypic characteristic are used at genetic or molecular level to characterize the organism. Thus include a nucleic acid probe, DNA amplification, rRNA sequencing, and essentially PCR or polymerase chain reaction.